Vice Chair of Research Integration and is the Director of Center of Statistics in Big Data. Dr. Li has been elected as a Fellow of the American Statistical Association, a Fellow of the Institute of Mathematical Statistics, and a Fellow of American Association for the Advancement of Science. He has served on the board of scientific counselors of the National Cancer Institute of NIH and regularly serves on various NIH study sections. Furthermore, Dr. Li has served as chair of the section on statistics in genomics and genetics of the ASA and co-editor-in-chief of statistics in biosciences. Dr. Li's research focuses on developing statistic and computational methods for analysis of large-scale genomics and metagenomics data and theory on high-dimensional statistics. He has published more than 300 papers in renowned journals such as Science, Nature, JASA, Biometrics, among others. So today he's going to talk about regressing Gaussian distributions on vector covariates for single-cell gene co-expression analysis. Let's welcome.
Thank you for the kind introduction. So I'm an applied statistician. I feel really honored to give a talk at this conference of basic science. So I'm going to talk about regression Gaussian distributions on vector covariate for single-cell gene co-expression analysis. And this talk is based on joint work with my postdoc, Arnaud Adi, my PhD student, Hongzhe Xu, and my colleague, Tony Chai. So the motivation of this research is analyzing the population scale single-cell RNA-seq data. So the idea of population scale single-cell data is that we have a sample, a collection of sample individuals from a population. And those individuals, we may have their individual specific covariate or disease status. And in addition, for each individual, we have collection of single cells, usually range in terms of couple thousand, 5,000 single cells. And, different individual, you may have different number of cells. And for each of the cell, you have a measurement of the expression of the genes. So this sort of single cell data really allow us to estimate the distribution of a set of genes for any given individual. So each individual, because we have thousands of cells, we are able to estimate the distribution of gene expression, or either one single gene or a set of genes. So the question we are interested in answering is, how does this distribution, gene expression distribution, change with time or with a covariate? And I think in the next couple of years, we're going to see a lot of this type of population scale single cell data. This is just one example. This is one ongoing cohort study. For this particular study, they're going to collect single cell data of the blood PBMC cells over 10,000 individuals. That include about 5,000 healthy individuals and a couple thousand people with different diseases. And for each of the individual, they're going to measure the expression of the genes of roughly 5,000 cells. And I think we're going to see a lot of data like this. So the question is this. As I mentioned that because every individual, you have so many cells, for a set of genes, you can look at the distribution of the expression over the cells. So statistically, suppose we have NIID samples from a pair of random variables, x and y. The x is a covariate sitting in the p-dimensional Euclidean space. So p-dimensional covariate. The y is outcome, and we assume y sits in some general metric space. So y could be a distribution of the gene expression, or could be the covariance matrix of a set of genes. And x are the predictors or the covariate. And because y belongs to a metric space, usually you need to introduce a distance of two elements of the y. And after you have the distance, the standard way to define the mean for the metric space is this Fauché mean. which is the y which minimize the expected value of the square of the distance. And the extension of the Frechet mean is a conditional mean, which is a regression. This Frechet regression model was proposed by Peterson and Mueller in 2019. So their idea is we're going to estimate the conditional expectation of the y given the x, given the covariate being the little x. And this conditional mean is defined by the y that minimize this weighted square distance. And they pick this particular weight. So this weight depends on the covariate x, observed covariate little x. And particularly, they define the weight in this particular form, with x is our covariate, mu x is expected value of x, and sigma x is covariance matrix. So this is all defined by the Euclidean vector, standard Euclidean vector. Which one? The third line. Oh, yeah, we're going to define. This is in general. So, for particular distribution, on next page, I'll tell you what is the definition. So this is in general setting. For any distance for the metric space, you can formulate the regression this way, right? And the reason they defined this weight by this particular form is if the y is the standard scalar outcome, and if the distance is a standard square distance, then the Frechet conditional mean defined this way is actually reduced to the standard linear model. That's the main motivation. So you do a little calculation, you will see, suppose d is a square distance, then this conditional expectation is exactly can be written as ax plus b. So that's their main motivation. Now, to answer this your question, usually when we consider two different distributions, either one-dimensional distribution or multi-dimensional distributions, one way to define the distance is the Wasserstein distance, and which is defined by, so you have two probability distribution, mu1 and mu2. Wasserstein distance is defined by the inf over all the coupled distribution, all the joint distribution with the marginal distribution specified by mu1 and mu2, and expected value of the square of the distance. So this is how they define the distance, the d, the distance d, a particular Wasserstein distance. This distance is, in general, is very difficult to calculate, right? But if you have a Gaussian density, for example, if you have two multivariate normal distribution, actually there's a closed form solution for this distance, which can be written as this. The first part is the difference of the mean vector. The second part is the difference of the covariance matrix. So this is very nice if you have a normal distribution. So particularly for the single cell data, we are really interested in answering the question of, does the gene co-expression depend on some covariate? In a particular example, I'm going to show you on age. And gene co-expression can be summarized as a covariance matrix of a set of D genes. So every individual, you have single cell data, and you select D genes, you can compute the covariance matrix. I'm going to call that covariance matrix as a matrix for co-expression. Okay, so the question I want to answer is, does this gene co-expression depend on covariate, such as age? For now, just consider the ideal setting, and you will see later on, I'm not going to need the assumption of normality assumption. But suppose for each of the individual, we observe a distribution, we know a distribution associated with each individual. Remember, we can estimate this distribution based on the single cell gene expression data. Okay, so every individual, you have their own covariance matrix. We really just care about the covariance matrix, and we don't care much about the mean. So the null hypothesis of interest is whether this is sigma I depend on the covariate or not. That's the null. And I'm going to develop a test statistics based on the Frechet regression. And the idea is quite simple. Basically, you can just compare the Frechet mean, the covariate specific Frechet mean. The Frechet mean you estimated for a particular covariate you observed for the IC individual with an unconditional Frechet mean under the null, right? Under the null means the mean does not depend on the covariate. So you just basically compare how different they are. Yeah.
You are interested in co-expression because you want to see the interaction?
Yeah, basically this whole matrix, whether there's association with covariate as a whole. Yeah, not a particular pair, not the individual, global test. Yeah. Okay. Yeah, feel free to interrupt, ask questions. Okay. Now again, let's go back to the ideal setting. Suppose we have n independent Gaussian distributions. Remember, our y is a collection of distribution. For now, it's assume a multivariate normal distribution. Then for each of the individual, we have the weight. Remember, this weight is defined by the covariate for each of the individual. Then we can define something called condition. Remember that the Frechet regression essentially is a conditional Frechet mean, conditional on covariance, defined through using this weight, that particular weight I just mentioned. And it's turned out the Frechet mean of the Gaussian density is another multivariate normal distribution. And with the covariance specified with the conditional mean, with its mean now is a covariance matrix, right? That covariance matrix can be solved by this fixed point equation. Iteratively, you can solve this equation to obtain the covariate specific covariance matrix. And later on, I'll show you how to solve this equation by using some version of the gradient descent algorithm. You can efficiently solve this equation. And after you have the mean, basically, we want to perform a test basically under the null if there's no effect of the covariance on the distribution, that essentially saying the Frechet mean, the conditional Frechet mean, is the same as the Frechet mean under the null, right? And so one way to construct the test is, so let me just clarify what this is. So this term, the first term is the multivariate normal distribution estimated by the Frechet regression, condition on the covariate for a particular observed covariate for the IC individual. And this is the multivariate normal distribution under the null. And you just calculate the Wasserstein distance between those two normal density and sum them together. So under the null, this distribution should be close to this distribution. Therefore, the distance should be small. So under the null, this F statistic should be small. That's the idea. However, we don't really observe the true distribution of the true covariance matrix for each of the individual. In practice, we're going to use a sample version. So for each of the individual, based on the single cell data, we can estimate the covariance matrix and calculate the Frechet mean, the covariance specific Frechet mean. Use a sample version to construct the statistics F star hat now. So hat means we're going to plug in the sample estimate of the covariance matrix. Now, when you study you try to study the limiting distribution or the asymptotic distribution of this test statistics, you need to remember there are two sources of randomness. One is that we have n IID sample from population. Those are the n individuals from the population. So that's one randomness. Another randomness is for a given individual, we have a sample of single cells, so another source of randomness, right? We're going to use, for each individual, you're going to use the M1 number of single cells to estimate the covariance matrix. So when you derive the limiting distribution of this test statistics, you need to consider two types of randomness. And we all know estimating the covariance matrix in relatively high dimension setting often is difficult. It's not that easy. The sample size requirement usually is large. So we consider one particular setting, which we assume for each of the individual. Remember that sigma I is a true covariance matrix of a set of gene expression for the IC individual. You're going to assume the so-called common PCA assumption. Assume the covariance matrix from individual, they share the same eigenvectors, the same PCAs, the same eigenvectors, but they have individual specific eigenvalues, lambda I, okay? And again, I'll show you later on with some real data. You see, this is actually a reasonable assumption. And later on, I'm also going to relax this assumption also. For now, just assume the covariance matrix, they share the common eigenvalue. If you look at the single cell literature, often you see this type of a t-SNE plot. That is quite reasonable assumption. They assume people, they share the common PCs. The only difference among different individuals are those lambda I. So this is nice because now we can estimate the U by using the single cell from all the individuals. And for the typical study, the number of cells is around millions. So if you have millions of single cells, you are able to estimate this U matrix very well because the sample size is extremely large. Because you're going to aggregate the cells across many individuals. And after you have the U, you can estimate individual specific eigenvalues. You don't need a lot of samples. So that's basically the idea. So in this case, the Frechet mean has this very simple expression, which is the conditional Frechet mean from the Frechet regression. Basically, can be written as this formula, where the covariance has effect on the eigenvalues, not on the eigenvector. Okay. So let me say, I mentioned this assumption makes some sense biologically. For example, there's 3 different ways to understand this assumption. One is this assumption basically assumes the same independent factors or independent source of variation in the single cell data for all the individuals, but with a possibly different variance that depend on the covariance. So the same independent source of variation of the observed single cell data for each of the individuals. Or those PC can represent a gene expression program that are consistent across all the individuals, but with an individual specific weight. Another interpretation of the PC are in genetic literature, they call the eigen genes. And basically, we are modeling the variability of the eigen genes. And another thing I want to mention is when we try to model the covariance as an outcome, then it's much more complicated than try to model the mean. Because the mean, you can regard the mean to be sitting in the Euclidean manifold. But when you look at the covariance, they sit in this curvature, in this Riemannian manifold. It's not in the simple Euclidean space. So we need a new tool to model the outcome sit in the particular manifold. That's the key of our approach. Okay, so under this assumption now, if we assume the true covariance matrix has this common PCA structure, then we can derive the test statistics now in this explicit form, where the first part, so the P is a number of covariates, D is a number of variable, number of cell, number of genes we are considering. And this nu is related to the eigenvector of the covariance, of the covariate, related to the covariate. Lambda are the eigenvalues for each of the individuals. Okay, so you can write down the test statistics in this closed form. So this is, again, assuming if we actually observe the covariance matrix for each of the individual. But as I mentioned, in practice, we only observe the data sampled from a distribution. Right? For the IC individual, we have MI cells, and use the data observed over this MI single cells, you can estimate the covariance matrix. And so the way we do this, as I mentioned, is we're going to split the data into 2 halves. Use the first half of the data, aggregate all the cells, divide into 2 parts. The first part of the cells, we pull all the data together to estimate the covariance matrix of a set of genes. So this sample size can be very large, 500,000 to 1 million, very, very large. So we can estimate the sigma hat very accurately. After you estimate the sigma hat, you can do the singular value decomposition to obtain the eigenvector. And after you have the eigenvector for each individual, you can estimate individual specific eigenvalues. And you plug in those eigenvalues to get the final test statistic in this form. Okay, so it's not too difficult to calculate this one. Okay. Now, the limiting distribution, in order to perform the statistical test, we need to derive the asymptotic distribution under the null. So again, under the null hypothesis is the covariance matrix has the same distribution. This sigma I does not depend on the covariate xi. And the null distribution can be written as a sum of weighted sum of independent chi-square 1 distribution. And the number of the components, number of chi-square 1 component is d by p. D is the number of genes we are considering. P is a covariate of the X that you want to test for association. And this limiting distribution, this is the weight will depend on those quantity. The ZX are observed. Those are the you can estimate ZX matrix by observed covariate. The D matrix depends on expected value of those eigenvalues, but this can be estimated by the sample average. So all those terms can be estimated to perform the test. Okay. So I just want to point out a few features. So the asymptotic null distribution, when we derive this reflects two sorts of randomness because we requires the number of samples to be bigger than the dimension, number of genes you are considering. And for each individual, the number of cells need to be bigger than the dimension because we want to use the certain number of cells to estimate the covariance matrix. But we don't really, the whole derivation of this asymptotic distribution does not need the normality assumption. It is no normality. I just use a normality assumption to motivate the statistics because for normality, multivariate normal distribution, we have a nice formula for the distance, for the Wasserstein distance. But all you need are the covariance matrix. No assumption on normality of the data. And the asymptotic result works if the eigenvector of the z's, this is only dependent on the covariate matrix, is incoherent with hold for the random design. So basically, it's just say there's no one single vector that are dominate other eigenvectors. So this is a test, and we can also derive a similar test to test for the partial association or a conditional association. So suppose your covariate, you can divide your covariates into two parts, x1 and x2. You can test the conditional independence of the sigma with x2 conditional on x1. For example, in biological application, in order to test the association between the covariance of a set of genes and a particular covariate, you might want to adjust for other covariate. In that case, you perform this conditional test, and you can derive a similar statistics here. Okay, so let me just show you some simple simulation results just to demonstrate that the test statistics indeed follows the null distribution with a mixture of chi-square distribution. So this is a setting where I consider 50 dimension 50 genes, 2 covariates, a total of 800 people. For every individual, number of cells is 200. And this figure is under the null. Basically, we can simulate some true covariance matrix and generate some data. So the first part is every individual have the same covariance matrix, and this is QQ plot of the theoretical quantile and the empirical quantile. You can see the empirical quantile follows the theoretical quantile closely. The second plot shows the power of our test as a function of the effect size sigma. So the second setting, I'm going to assume the eigenvalue, see here, the first and second eigenvalue depend on the covariate. And you increase the coefficient delta, you see the increase in power. So basically, it behaves reasonably well. for this setting, typical setting, single cell study, population-based single cell study. Number of cells 200 is usually small. Usually for typical study, number of cells could be in the order of a couple thousand. So now back to some of the data applications. So there was one paper published in 2022 in Science. In this paper, they look at the single cell gene expression of the PBMC. This is blood cells. PBMC cell derived from the blood of 982 healthy individuals. And those 982 individuals are the sum of individuals at different ages, from 20 years old to 80, 84, 85 years old. And particularly, we look at 61 genes that belong to a particular nutrient sensing pathways. And this pathway is related, agent is shown in literature to be associated with the deregulation of nutrient sensing pathways. So it's a total of 61 genes. And single cell data also allow us to really focus on a particular single cell type. And in this case, we look at a particular cell type called CD4 T cells, which is also somehow related to aging. So out of this 982 individuals, there's some individuals you only have a small number of cells. We remove them. So this is the number of cells we're going to use to estimate the covariance matrix of a set of genes. And 61 genes, only 26 genes are kind of expressed in those in this particular type of cells. So 26 genes, they are expressed in at least 3% of the cells of this particular type of cells for each of the individuals. So eventually, we just look at the covariance structure of this 26 genes, and we want to see whether the co-expression change with the age. Okay. Yeah.
Different cell type, you concentrate on those cells.
Yeah, a particular type. So that's reduced the number of cells quite a bit. Otherwise, in average, we have about 3,000 to 5,000 cells for if you look at all the cells. But I think in this case, yeah, I think both. You look at all the cells, look at the co-expression, also tell you something, or a particular cell type. People do the co-expression, looking at the bulk RNA, essentially look at the aggregate of all the cell expression. So, but for this particular analysis, let's look at the CD4 T cells. Okay, so I mentioned the assumption. Remember, to derive the limiting distribution of the test statistic, I'm going to assume every individual shares the same eigen vectors. And that assumption, particularly for this data set, actually works pretty well. So what you see here are the top 3 eigenvalues. So this is the eigenvalue for each of the individual. Each dot represents one individual. And this is an estimate, 2 different estimates. The estimate you use, so each individual, you estimate the covariance matrix, then you calculate the eigenvalues. And another estimate, you assume they share the common eigenvector, then you estimate the eigenvalues. Turn out that the estimate of the eigenvalue are quite similar with or without assumption of common eigenvector assumption. So that indirectly indicated that assumption may not be a bad assumption for this particular data set. So here are the plot of the first 2 eigenvalues for each of the individual. This is age, as I mentioned, ranges from 20 to 80 some years old. Most of people are older than 55 years old. And there are 2, so there, what I plot is the eigenvalue observed, estimated eigenvalues. There are 2 lines. One is the blue line is just a simple low S fit, and the red line is our estimate. Remember, you run the first regression, you can estimate the eigenvalue as a function of age. This looks like a linear. It's not quite linear. It's just the change is little gradual change, but it's not quite. You cannot show this is linear, even for one single covariate. So if you apply our test, we can get a p-value of 0.03, in the sense indicating there is an association between the covariance of that small set of genes and the age. But another interpretation, remember I mentioned this PCA can be interpreted as eigengenes. This, I think, essentially is saying as people getting older, this set of eigengenes, the variability getting bigger and bigger, right? And this actually, there are some interpretation of this result. So I just mentioned why we want to look at CD4 cells, and there's literature indicating that this particular, as we age, this particular cell type will change. And the basic idea is this accumulation of highly differentiated T cells will change more as people getting older. So to some degree, I think by looking at the eigenvalues, we capture that variability of the eigengene as people getting older. So this is interpretation, again, is at the global summary instead of a co-expression of a pair of genes. So this is basically, I think this single cell data is really useful in terms of providing a distribution of the genes across cells. So this enables us not to just look at the cell type specific mean gene expression, which is typically what people do. They divide the cells into particular cell type and look at the mean expression of the genes. Actually, we look at those 26 some genes. If you look at their expression, not only you see a variability in terms of mean from individual to individual, there's also a large variability in terms of the variance of those genes for the cell of the same type. So it's not just the mean difference from individual to individual, there's a difference in variance. So this motivates us to look at the whole distribution. And since you have so many cells, you are able to look at the distribution of the genes across cells for every individual. And if you look at the distribution, so we have a way of studying how the distribution change as a function of the covariate. And if you only care about the covariance, then for say regression under Gaussian assumption, it leads to look at the covariance. But the Gaussian, again, I want to emphasize the Gaussian assumption is not required. All the proof, you don't need a multivariate normal assumption. So we derive a test under this common PCA assumption. That's the key assumption. We assume every individual covariance share the same eigenvector. The only difference are the eigenvalues. So this is the first part of my talk. We have a paper on this is under review. Now I want to talk a little bit about my recent work with a student in applied math program at Penn. So I want to relax this assumption because this is not very, it seems to be a reasonable assumption for the data we look at, but I want to relax this common principle, common PCA assumption to consider a general regression setting where I have covariance matrix as outcome and want to regress on a set of Euclidean covariate. Okay. So here is a similar setting now. So what are the data we have? For now, we assume we have n individuals, IID sample. X is a covariate in the p-dimension Euclidean space, and Qi is a positive definite covariance matrix. And again, for now, I'm assuming we observe this covariance matrix, and later on, I'm going to show you how to replace this Qi with an estimate from the data from the sample. Okay, so I have IID. My outcome is a positive definite matrix of dimension d by d, and my predictor is Xi is a p-dimensional covariate. And again, for this positive definite matrix, there's a nice distance that we can define between two positive definite matrices, and this distance is called Wasserstein distance. This is exactly the same as the distance between two multivariate normal distribution if the mean is 0. Okay, so now you have a particular space of positive definite matrix, and you have a distance. Just like what we did, we can define the Fauché regression as a conditional mean of the covariance matrix, which is defined by the covariance matrix that minimize the expected value of this weighted distance. Again, this weight S is defined by the covariance, by the covariate X, the same as before. And we can, so this is a theoretical model, this is a population model. And in practice, we can estimate this conditional mean by using the sample average of the weighted distance. And I'll show you the algorithm to do this calculation. Again, our goal, the null hypothesis, is we want to see whether this covariance matrix is dependent on the covariate X. Whether it's under the null means the covariance matrix is the same for all different X. Okay, so that's the more general setting. Okay, now this is more involved. To get the test statistics and the limiting distribution under the null is a lot more involved than the previous approach, because previous approach, all you care about are those eigenvalues. So some of the tools from optimal transport literature I found is very interesting and very useful. So I list some of the key facts from the optimal transport literature. So the idea of optimal transport is for example, if you consider two distribution, multivariate normal distribution with mean 0, variance covariance matrix Q, and variance covariance matrix S. Remember, Wasserstein distance essentially try to find a transformation to map from one distribution to another distribution that minimize the cost. The cost is defined by the L2 distance of the two random variable. And it's turned out for the Gaussian distribution, there is this closed form expression for the optimal transport map from one covariance matrix to another covariance matrix. And this is called the if you consider two distributions, this is called push forward between two distributions. You push one distribution, the random variable from a random variable from one distribution, you apply this map, and you get a random variable in another distribution. But I don't need a normality assumption again, right? Because for two matrices S and Q, I can just define the optimal transport map this way. Okay, so you can regard this as a function of Q for a fixed S. And as a function of Q of fixed X, you can talk about differential derivative or differential or Fauché differential. So this is another really nice property is this map as a function Q with respect to Q for each fixed S is a differentiable as defined into this general formula, Fauché differential formula. Okay, so this transport map at the QQD equals the transport map at Q plus this differential operator applied act on the difference of QQD and Q. And there's a 2016 paper, you can derive a closed form expression of this differential of optimal map. It's a very nice result. Is basically, if you give me this term, you can derive closed form solution of this differential operator. Okay, so those are the two key tools that we use in derive the test statistic for testing the null hypothesis. So the test we came up with has this nice formula. So again, the null hypothesis is whether the covariance matrix does not depend on the covariate X, p-dimensional covariate X. And the test statistic you can think about, so what is this term? This guy, QNX bar, is the estimate of the Fauché mean, the average of the covariance matrix under the null. And the first one is, remember the Fauché regression gives us the covariance specific estimate of the covariance matrix. So this is estimate from Fauché regression, this term, which depend on the covariate xi for the IC individual. And so again, in a sense, those under the null, those two covariance matrices should be small. But in order to derive, so you can see, okay, why don't we just take the previous Wasserstein distance between those two estimated covariance matrix and define a test statistics that way. You can do that, but it's very difficult to derive the limiting distribution, to get the asymptotic distribution of that. So basically the idea is those are the difference. Remember the walled test, if you think about generalized linear model, you want to test whether beta is 0. What you do is you can estimate the beta under alternative, under the beta, under the null. Then you look at the difference of the two beta, and you look at the difference and you create a score weighted by the variance of the estimate. That's a typical wall test, right? So it's theta hat minus theta hat under the null times the inverse of the covariance matrix times that. Exactly the same idea. So you take the difference here of the estimate under the alternative and estimate under the null, and H is the differential. This is what I mentioned, this Fauché differential operator. Remember, this optimal transport essentially is a gradient of the it's a gradient in this manifold. You take a further derivative of the gradient, essentially second derivative, give you a sense of the variance of your estimate. So you need to multiply this H hat, not multiply. You need to apply this H hat differential operator to this difference, and you take a Frobenius norm to define the test statistics. So this is how we define the test statistics. Turns out the limiting distribution is not that easy to derive. This, if you look at our, we have an archive paper if you are interested in. This cost you a long time to work this out. This archive paper has 98 pages. Just try to figure out the limiting distribution of this test statistics. But it's really make intuitive sense. So this is the final limiting distribution under the null. It's again, it follows a mixture of chi-square distribution, independent chi-square distribution. But now it's instead of chi-square 1, it's a mixture of chi-square p distribution. P is the number of the covariate you want to test. I'm sorry, violate what assumption? Correlation of gene expression? Where? So maybe it's because so far, so for now I assume for each individual I observe this covariance matrix. Then I'm going to relax this assumption. So basically I want to every individual you have observed you have the true covariance matrix for now for a set of genes. Then later on we're going to have data, single cell data to estimate the covariance matrix. So this result is based on the fact that I assume I know the covariance matrix QI for the IC individual. Okay. So the test actually is really easy to implement because this transport we have a closed form solution, and this is identity operator. And you can just replace this expectation with a sample average across any individuals. and you can perform the test. Now here's the algorithm, how to actually implement the first-hand regression. It's a very nice algorithm. There's a lot of new literature, new things on this optimal transport, Wasserstein distance. So this is a nice algorithm published in 2020. They call it Riemannian gradient descent algorithm for the first-hand regression. So the idea is it's very, very easy to do, right? So the input, we have NIID individual, and we have covariate, which is Xi for the IC individual. The response are the covariance matrix. For now, just assume I have a covariance matrix Qi. And you can start with, so remember the first-hand regression, essentially we want to estimate the conditional covariance matrix, conditional on the X. So you have to start with some initial value. The idea is you can update the, you can do the gradient descent in this tangent space of the manifold. Then after you update in the tangent space, you map back to original manifold of the positive definite covariance matrix. And that in the tangent space, this guy essentially is a tangent vector. This guy, this is a transport minus identity operator. And so those are just a matrix operation. And you can update you do the gradient. So this is a tuning parameter in the gradient descent in the tangent space. And after you update the G, all you need to do is you map it back to the original covariance matrix. So it's, actually it can be done very efficiently by using this algorithm. Now I mentioned that in practice, obviously, we don't observe the Qi. What we observe is the estimate of the covariance matrix from the data. So the data we observe is a Qi tilde. Tilde is the estimate of the Qi. And what we can show is as long as your estimate is decent, for example, your estimate needs to be unbiased, and the error of the estimate is controlled by something like n to the minus beta order. N is a sample size, number of individuals, and beta is some constant. Usually it's about 1/4 of the constant. So this condition hold if you truly have a multivariate normal observation, and you just do the sample, use a sample covariance estimate as the Q hat, then this condition hold. So in any case, if you have a decent estimate of the covariance matrix, then the idea is you just replace the Qi with your sample estimate, and the test still performs well. So the thing is that the M in our case is the number of cells for each individual. It has to be bigger than square root of n. It's the number of samples, but can be much smaller than n. You don't really need too many cells in order for the test works in this case. So I just want to show you some simulation results, for example. So here is the setting where I have 10 genes, so D equals 10, number of individuals 200, and this plot is a power curve based on 200 replications as a function of the effect size delta. So basically, I assume the covariate X affects the covariance matrix by this simple function. And I draw observation from a multivariate normal distribution, and I estimate the covariance matrix by the sample average. And you can see the power are similar. Really, you don't really lose a lot in terms of power as you reduce the number of cells. The M is the number of cells. M equals infinity means we know we observe the covariance matrix. M equal to 1,200 to 100 are the number of cells, number of samples you have to estimate the covariance matrix. You lose the power a little bit, but not very dramatically. In terms of the type and error, again, this is the QQ plot of the theoretical quantile of the null distribution and the empirical quantile of the test statistics. The first scenario is the setting where I assume I know the covariance matrix. Every individual, I observe the covariance matrix. The second scenario is I plug in the sample estimated covariance matrix. And you can see for M, its number of cells is 200. You can see the test statistics still follows the right distribution. Even for, in this simple setting, for a number of cells of 200. So I think if you have a couple hundreds of cells, you probably, for a small number of genes, you cannot look at a couple thousand genes. If it's 2,000 genes, the covariance matrix is 2,000 by 2,000 divided by 2. That's a very large matrix. You need to have a lot of cells in order to get a decent estimate of the covariance matrix. But if you have a relatively small set of genes, it's probably doable. So we apply this general approach to the same data set. Again, this is the estimate of the top 4 eigenvalue as a function of age. You can still see for all 4 of them, you see there's a monotone increase trend over time as people getting older, the variance of those eigengenes getting bigger. And use this test, we got a p-value of 0.0012, indicating a significant association between the covariance matrix of that set of genes and the age. So in summary, I just say that this paper, again, if you are interested in, it's available on arXiv. Essentially, in our paper, we derive a couple of things. One is a new test statistic for testing the association between covariance matrix and a Euclidean covariate. And in order to derive the asymptotic distribution of this test statistics, we need to have the uniform convergence result of this regression model. This is regression. This is a conditional Frechet mean of the covariance matrix. And we also, in our paper, we also derived the pointwise central limit theorem for the regression estimate of the covariance matrix. So in summary, this is something my group in the last couple of years started to look at this population scale single cell data analysis. We're really interested in understanding how the single cell measurement variability across many different cells for each individual, how they are related to individual specific covariate. I think this Frechet regression framework provides a nice framework to associate the distribution of a set of genes to individual specific covariate. And my group also are working on quite a few other potential applications of optimal transport for the population level single cell data analysis. I think that's all. Thank you very much.
Okay.